When gametes are formed in the process of meiosis, crossing over occurs between homologous chromosomes. Usually, crossing over involves comparable regions. However, there are cases where chromosomes do not align perfectly and unequal crossing over occurs. In such a scenario, one chromosome, such as the one in the upper right, can end up with duplications of genes.
That's it. I just want to show a visual representation of this crossing over. Evolution of genes with related functions, human globin genes. Duplication events can lead to the evolution of genes with related functions, such as those of the alpha and beta gene families, which in the next slide I'll show you a chart of those. And this approach to recreating evolutionary history of the globin genes indicate that they all evolved from a common ancestral gene that underwent duplication and diverged into the alpha and beta ancestral genes. Here's this chart, and it is believed that this divergence from the common ancestral globin gene occurred about 450 to 500 million years ago, and gradually more and more duplications and mutations occurred. Each of these was duplicated several times, and this makes a type of globin superfamily. The differences between these genes arose from mutations that accumulated over many generations. An important concept to realize is that though some mutations had no effect on the organism and some had an adverse effect, a few mutations must have altered the function of the protein in a way that was advantageous to the organism. Evolution of genes with novel functions. Gene duplication and subsequent divergence produce family members whose protein products perform similar functions. An example of this is lysozyme versus alpha-lactalbumin. Lysozyme is an enzyme that protects animals from bacterial infections, and alpha-lactalbumin plays a role in milk production in animals. Since they came from the same ancestor, there are many similarities. Two very different functions, but they are quite similar in amino acid sequence and three-dimensional structure. Both genes are found in mammals, while only the lysozyme gene is found in birds, which suggests that at some time the lineage of mammals and birds had separated. This suggests at some time after the lineage of the mammals and birds had separated, the lysozyme gene was duplicated in the mammalian lineage but not the avian lineage. So as Sam mentioned earlier, with transposable elements, they are defined as a DNA sequence that can change its position within the genome, sometimes creating or reversing mutations. Most times these are harmful, but in the long run may prove beneficial by providing a survival advantage. Each transposable element can carry a gene or a group of genes to a new position on the genome, which provides genetic diversity and a valuable raw material for natural selection.
Comparing genome sequences. Comparisons of genome sequences from different species reveal much about the evolutionary history of life, such as one species evolved into another one. The more similar in sequence the genes and genomes of two species are, the more closely related those species are in their evolutionary history, such as comparisons of closely related species, such as humans and chimpanzees. It sheds light on more recent evolutionary events, or comparisons of distantly related species, such as humans or yeast, and it helps us understand ancient evolutionary history. Analyzing highly conserved genes in distantly related species can help clarify evolutionary relationships among species that diverged a long time ago, such as comparisons of the complete genome sequences of bacteria, archaea, eukaryotes, that these three groups diverged 2 to 4 billion years ago. And genes that evolved a very long time ago can still be surprisingly similar in disparate species, such as several genes in yeast that are very similar to human disease genes. Genomes of the two that are likely to be organized similarly due to recent divergence. It allows fully sequenced genomes of one species to be used as a scaffold for sequencing the genome of a closely related species, and it underlines the small number of gene differences. The particular genetic difference then can be more easily associated with phenotypic differences. Comparing the human genome with the genomes of the chimpanzee, mouse, or other mammals gives insight in what it takes to be a mammal, a primate, or more specifically a human. And an analysis of the overall composition of the human and chimpanzee genomes, which are thought to have diverged 6 million years ago, reveals some genetic differences. Considering single-base substitutions, the two genomes only differ 1.2%, which isn't a lot at all. But then further research revealed in longer stretches of DNA that there was a further 2.7 difference. This is due to insertions or deletions of larger regions into the genomes of one or the other species. And all these observations provide clues on what might have swept the two genomes on different paths. Studies of specific genes and types of genes that differ between humans and chimpanzees and comparing them with their counterparts in other mammals provide the basis for phenotypic differences. These have revealed a number of genes that are evolving faster in humans than in a mouse or chimpanzee, such as genes involved in defense against malaria and tuberculosis, and at least one gene that regulates brain size. When genes are classified by function, the genes that seem to be evolving the fastest are those that code for transcription factors. One transcription factor in humans that shows evidence of rapid change is called the FOXP2 gene, which codes for the vocalization in vertebrates. Mutations to this gene can cause severe speech and language impairment in humans. Researchers are trying to determine whether the differences in the FOXP2 proteins account for the ability of speech in humans, but not chimpanzees. There's only a 2 amino acid difference between the two, so it's a very small difference. And these studies are helpful for other human genetic disorders such as Down syndrome. And our ability to analyze genomes is increasing our understanding of genetic variation in humans. Because of the history of the human species is so short, only about 200,000 years. The amount of DNA variation is small compared to other species, and much of the human diversity seems to be in the form of single nucleotide polymorphisms. And then scientists have already identified the location of several million single nucleotide polymorphisms, sites in the human genome, and these variations will be used as genetic markers for studying human evolution and for identifying genes that cause diseases in the future.
Here's our highly anticipated review game. Maggie, what is the genetic material of an organism called?
DNA? No.
No.
Wait. Niacin?
You guys gotta speak up when you answer.
Genetic material. Yeah, complete. Sam?
Genome?
Okay.
Good. And what is this study called? Tony?
Genomics.
Last question. What occurs when specific proteins are broken and joined to separate chromatids? Jackie?
I'm not sure.
Clark, do you know?
Is it?
Yeah, crossing over.